Hi friends, welcome back to our YouTube channel. If this is your first time coming across our video or visiting our channel, please make sure you like, share and subscribe. In this video, I'm going to talk about digestion and absorption of protein. And by now, I know you guys actually understand what digestion is and what absorption really means. So when it comes to digestion is just basically a process that a complex biomolecule or a complex molecule undergo for it to be converted to its simpler form. That is a step-by-step breaking down of complex molecule into its simplest form. You understand? And it's basically an enzymatic process. Now, when it comes to absorption of molecules, we are basically talking about the rate, or let me say, the availability of the simpler form of this molecule into the systemic circulation. That is how the body absorbs this simpler form of the molecule, right? So now, basically, in this video, I'll be talking about the digestion and absorption of protein, right? And we know that protein are polymer of, let me say, protein are polymer. They are polymers of amino acid. Amino acid, that is, they are formed from the joining together of amino acid. That is, when two or more amino acid, or let me say more than like many amino acid actually joins together, right, through peptide bond and different kind of bond. That's basically what protein is. So protein are polymers of amino acid, while the amino acids they are the monomers of protein. So in this case, we are basically breaking down. We are going to be breaking down the protein, which is the complex form of amino acid. We break it down into amino acid. Then the body is going to absorb the amino acid, which is the simpler form. That's basically everything that this video actually entails. Now let's just start from here. Now, first thing that I want you guys to understand is that this diagram that you're seeing here actually represents the gastrointestinal tract. That is, it represents organs, the system, and also the cells, the tissue that are responsible for the breaking down of biomolecule into its simpler form. Right? Now, from here, this particular portion that you're seeing here actually represents, let me say, this represents the oral cavity, right? This is the oral cavity, as you can see, right? So this is basically where we are going to ingest our food. And this particular gland that you're seeing here is called the salivary gland. Then this particular narrow shape that you're seeing here is basically the esophagus. Then this diagram is the liver, right? Then we have our stomach here. This is our stomach. Then from there we have the intestines. And don't forget that the intestine is made up of three parts, right? We have the first part, which is the first outer part, which is the duodenum. We have the inner part, which is the jejunum, and the last part, which is the ileum. Right? Now, if you look at this very well, we have different kind of cells here, right? These cells are basically epithelial cells. They are basically the cells of the intestine, which is also referred to as the enterocyte. Right? So now, if you look at this, all of them are made up of. They are just like they are arranged in a finger-like structure, like a finger-like shape. Right? We have one cell beside another one, like they are arranged beside each other. And in that case, if you look at this particular small molecule or particle that you're seeing here, this small particle is basically is basically called the microvilli, right? While a single cell of the intestine, right, of the inner intestine, or let me say a single cell of the wall of the intestine, which is the lumen, is referred to as enterocyte, right? So this is a single enterocyte. This is another enterocyte. But this shape that you're seeing here, they are basically called the microvilli. The microvilli. Right, they basically help in the absorption of food. That is, they provide large surface area for the absorption of food to take place. Now, basically, let's just come back here and talk about the digestion process before we move on to the absorption process. Now, first thing is that, let's say you consume a food that is very rich in protein, right? You consume a food that's very rich in protein. Let's basically say, for example, we have meat, right? You eat meat, we have fish. Then let's simply say a little bit of beans. Right? If you eat this food basically they contain what? They contain protein. They are rich in protein, right? They are proteinous food. Now let's say you basically ingest them, right? Now if you ingest them, what actually happens in the mouth is that, right, the digestion of protein doesn't take place in the mouth. So once you chew with your food, then you mix the food with the saliva. Then what we're going to have is that we're going to have a bolus. Let's say a bolus. Bolus is basically a food that is mixed with saliva. Right, then the bolus is going to pass down. Right, let's say this is the bolus, right? This is the bolus that you're having. Right, let me just simply put it here a little bit. Right, this is the bolus of food. Then once it's entered the stomach. Now look at what we have here. This is the bolus now. Now this bolus, let's basically say it is what is a protein. That these are protein because digestion of protein starts from what, from the stomach. Now one thing I want you guys to notice here is that once the food actually reach what to the stomach, right? There is there are these cells of what, of the stomach that are responsible in what, in the digestion of what, of protein, right? Because this food is all about protein, right? Now let's basically say I'm having some cells here, right? Let's say this is basically what the cells, and in this case I'm going to call these cells, I'm going to call them what, the chief cells of the stomach, right? These are the chief cells. They are chief cells of what, of the stomach. Then another one is that, let's say I have another cell here in the wall of what, of the stomach, still in the wall of what, of the stomach. Then I'm going to call these ones, I'm going to call them the parietal cell. I'm going to call them the parietal cell, right? Now one thing that happens is that in the stomach, these chief cells, they are responsible for releasing a specific type of enzymes, right? That act on what, on protein. This enzyme is called what? Is called the pepsin, right? Or you simply say pepsinogen, because one thing that happens is that pepsinogen is what, is the inactive form of what, of this enzyme. The inactive form of what, of this enzyme is what, is the pepsinogen, right? Then another one is that as far as parietal cells what is concerned, it is responsible for what, for releasing what, a special type of what, of, let me say hydrochloric what, acid. It releases what, hydrogen what, chloride, which is HCl, right? Now what will happen is that protein, they are basically what, they are basically a biomolecule, right, that has amino end, right, and also what, they have what, carboxylic end, right? So in that case, let me just simply say I'm having something like this. You understand? I'm having something like this. Then let's say we have some structure here, right? Then we are going to have different kind of amino acid that we have here. Then let's basically that this bolus is basically what we are seeing here. And again, you can see we have what, amino end, and we have what, carboxylic acid end. So this is basically what the structure of protein. Now what happens is that protein in the stomach is going to what, is going to break it down into what, into, let me say, different molecule, which are, we have the first one, we are going to have what, polypeptide, right? Polypeptides. We have what, we have peptones, right? We have, let me say, we can have tripeptides, right? And at the same time we can have what, dipeptides, and at the same time we can have what, let me say, that's basically what we are going to have. That's going to be what the result that we have here. So this is basically what, protein structure. Let's say we have protein here. Don't forget the bolus of what, of food is one that will reach this side. We are talking about digestion of protein. Now in this case we have what, we have the protein molecule, which is what we are saying here. Then we are basically going to break the protein down towards to all these products that you are seeing here, right? These are actually called what, they are called derived protein. Why? Because they are derived from what, from the digestion of what, of the proteins that we have here, right? Now let's go ahead and look at what's going to happen here. Now the chief cells release what, pepsinogen, while the parietal cell release what, release hydrochloric acid. Now what happens here is that pepsinogen is basically what, the zymogen form of pepsin, the inactive form of what, of pepsin. Now in order for the action of what, of this pepsin, or let me just basically say this is the inactive form, right? Inactive. Then we are going to basically convert it to active form, which is what, which is the pepsin, right? Which is the pepsin. And then I'm going to explain how it is being converted to pepsin in just a moment. Now look at this. What happened here is that protein, in order for the action of pepsin to be optimum on the structure of this protein, right, then denaturation has to occur in this case. And then what we basically cause the denaturation of protein is basically what, the hydrogen ion that comes from what, from the release of hydrochloric acid. So in short, parietal cell what, will release what, hydrochloric acid that helps what, to denature what, the protein. That is, it will unravel what, the structure of protein. Don't forget that what, protein is made up of let me see, four different types of structure. We have the primary, secondary, tertiary, and also what, quaternary structure, right? Now in this case, this particular protein structure that we're seeing here is a complex form, right? It has to be denatured, and then denaturation what will basically occur due to what, in the presence of HCl. Right, the parietal cell release what, HCl, that helps to denaturize what, the protein that we have here. It helps to denature what, the protein. Then once the protein is what, is denatured, look at the structure that we're going to have here. We're going to have something like this. Let me basically, okay, I'm still come back here. We are going to have something like this. Look at this. We are going to have amino end here, right? Then we're going to have something like this. We are going to have different kind of protein here, right? So we are going to have COO minus. Now look at this. We basically what, denature what, this protein that you're seeing here. through the action of what, of hydrogen chloride. That's hydrochloric acid. So hydrochloric acid helps what to denature what the protein. Then from there, the chief cells releases what, pepsinogen. So pepsinogen is what, is the key enzyme, key protein. Let me say a type of what, of protease enzyme in the stomach. Now pepsinogen is what, is in inactive form. It has to be converted what, to pepsin, which is what, the active form what, of the enzyme. So hydrochloric acid can basically help what, to stimulate the production of what, the conversion of what, of pepsinogen to what, to pepsin, by cleaving up the origin part, right? Then once it's cleaved up the origin part, the active side of what, of the pepsinogen is going to what, is going to be exposed. In that case, we call it what, we call it pepsin, right? So pepsin is basically act on what, on this protein structure that you are seeing here, then converting it to what, to polypeptide, peptones, and also what, tripeptide and also what, dipeptides. Now one thing I want you guys to understand is that this, after the action of hydrochloric acid, the pepsin itself can in turn stimulate its production. It can in turn act on what, on pepsinogen, convert pepsinogen to what, to pepsin, right? The process whereby pepsin act on pepsinogen and convert pepsinogen to what, to pepsin. That's basically what, autocatalysis. Now from there, the product that we are going to have is basically what, the polypeptide, the peptone, the tripeptide, and what, the dipeptides, as you can see now. So basically, what really happens in the stomach is that the chief cell release what, pepsinogen, while the parietal cell release what, hydrochloric acid. The hydrochloric acid helps what, to denature what, the protein, right? Then it also acts on what, on pepsinogen, converting pepsinogen, which is what, which is the inactive form of what, of pepsin. It converts what, to pepsin, right? Then pepsin can also stimulate what, its further like more production of what, of itself, right? So this pepsin actually helps what, to act on what, on this protein that you're seeing here, converting to what, to polypeptides, peptones, tripeptides, and also what, dipeptides. That's basically what happens what, in the stomach after the action of the hydrochloric acid and also the pepsin. I want you guys to know one thing like about pepsin. Pepsin actually require the pH of 1 to 3 to perform its optimum activity, because all enzymes actually have their own or their pH until they perform their optimum function. That particular pH is referred to as the optimum pH. Now the optimum pH for pepsin is 1 to 3. You understand? Now from there, the product of the protein that we have in the stomach is actually what, the polypeptide, the peptones, the tripeptide, and also what, the dipeptide. So these particular peptides we actually what, mix with the stomach acid content to form another kind of let me say, to form another type of products, and this is going to be referred to as chyme. You understand? So that shows that the bolus actually entered the stomach and leave as what, as chyme. Then once the chyme actually move and get to the let me say first part of the small intestine, which is the duodenum. Once the chyme reaches the duodenum, is basically what act on, let me say, it stimulates some cells like the S cells of the intestine, and at the same time we have the I cells of the intestine. Like we have the I cells. These are the I cells, and also we have the S cells. Now what happens is that the chyme is highly acidic, right? And at the same time, the chyme is the one that is responsible for stimulating, or let me say, for stimulating, like indirectly, the release of the pancreatic enzyme, right? Because this is the pancreas. The pancreas is going to release the pancreatic enzyme, which will later act on the chyme and convert to further products. Now from here, we have the I cells and also the S cells. The S cells basically secrete, let me say, they secrete secretin, right? Is basically an hormone. So these cells, both the I cell and also the I cell, they are basically referred to as let me say, enteroendocrine cells. Enteroendocrine in the sense that they are intestinal cells, and endocrine in the sense that they secrete their product directly into the blood. Now this is going to be the blood value that you are seeing here. Then from here, the S cell basically secretin. So the secretin will move into the blood, then travel to the pancreas, right? So once it gets to the pancreas, it's basically acts on a special type of cells in the pancreas. So I'm going to represent the cells. These cells are saying here, I'm going to call it the, let me say, pancreatic ductal cells. You understand? I'm going to represent as let me just call them pancreatic ductal cells. So and then these are the pancreatic ductal cells, and at the same time, the one that you are seeing here in black, they are basically called the acinar cells. You understand? They are basically called the acinar cells. Now, acinar cells, they are the ones that are responsible for secreting let me say digestive what enzyme. The pancreatic digestive enzyme is secreted by the acinar cells. Then the pancreatic daughter cell are basically the one that secretes a special type of molecule which we call let me say bicarbonate. You understand? Because in order for pancreatic enzyme to perform their optimal what function, they require pH that ranges between let me say their pH is basically 7 to 8. You understand now? So from there, this is kind of alkaline, right? Compared to the acidic content of the stomach. And we know that the food is coming in form of chyme. So once it gets to the duodenum, it stimulates the eye cells to release a certain type of molecule, which is actually called the cholecystokinin. You understand? Cholecystokinin, which is a special type of molecule. I'm going to explain its function. Then also it stimulates the air cell to secrete secretin. So this secretin will basically travel to the pancreatic daughter cell, right, and bind to the receptor on the cell, right, thereby causing the pancreatic daughter cell to release bicarbonate. Look at what we are going to have it on. We are going to have bicarbonate, which is ACO3 minus, right? So this is bicarbonate. Then from there, the eye cell as well, which is the red portion that you are seeing here, will also travel to the acinar cell. Once get to the acinar cell, it basically stimulates the acinar cell of the pancreas to release digestive enzyme. I'm going to represent that as DE. Right? And all these digestive enzymes, they require alkaline medium for them to perform their optimum function. And don't forget, the chyme is highly acidic. So this bicarbonate that is released by the pancreatic daughter cell will help to neutralize the acidic content of the chyme. So once it neutralizes the acidic content of the chyme, then the digestive enzyme will be available to perform their optimum function. Activate basically the cells of the let me say the air cells of the intestine to secrete secretin, we basically travel to the pancreatic daughter cell and cause the pancreatic daughter cell to release bicarbonates. So basically in the pancreatic daughter cell they release bicarbonate, while the acinar cell releases digestive enzyme, right? And CCK basically acts on acinar cell to release digestive enzyme, while secretin acts on the pancreatic daughter cell to release bicarbonate. So this bicarbonate function is to basically neutralize the chyme, and at the same time, so that the digestive enzyme that appears in the pancreatic juice can actually act on the chyme optimally. Then from there, another function of the CCK is that the CCK can also travel to the liver, right, and come to the gallbladder, causing the gallbladder to contract and releasing let's say it's going to release bile ducts, right? And that one we are going to explain that when you get to direction of lipid, right? So that's basically the function of CCK and also secretin. Now, one thing I want you guys to understand is that the digestive enzyme that appears in the pancreas, they are also present in their inactive form, right? And we have 4 of them, or let me say 5 of them. We have the first one, we have the trypsinogen, right? Then we have the second one, we have the chymotrypsinogen, right? Then we have another one, we have the proelastase. Then we have the procarboxylase, right? And this procarboxylase can be of 2 types. Let's say we have 2 types. We have the A and also B. We have procarboxylase A and procarboxylase B. That's basically what we have. So now, what happened here is that trypsinogen, chymotrypsinogen, proelastase, and procarboxylase A and B, all these enzymes are actually present in their inactive form, which is the zymogen form. Now, in order for them to be converted to the active form, there is a particular, or let me say, a special type of enterocyte here, enterocyte, which is the intestinal cell. These enzymes, this particular cell will release an enzyme, and this enzyme is basically known as enteropeptidase, or in some cases, some people just call it enterokinase. Right? Now we have it to be enteropeptidase, and what happens is that this enzyme basically acts on on trypsinogen, right? It acts on trypsinogen, right? And then converting trypsinogen into trypsin, right? It converts trypsinogen into trypsin. So trypsinogen is the inactive form, while trypsin is the active form. Now what happens is that this trypsin will in turn act on chymotrypsinogen. It will act on what? On chymotrypsinogen, and at the same time act on what? On pro-elastase, and at the same time it will in turn act on what? On pro-carboxylase. Also at the same time it's going to act on what? Okay, pro-carboxylase A and B. Yeah. So it's going to act on chymotrypsinogen and convert to chymotrypsin. We're going to have chymotrypsin. Right? And at the same time, the trypsin itself will act on what? On pro-elastase, cutting out what? The pro out and what? And we are going to have what? Elastase. Right? And at the same time, for the pro-carboxylase, we're going to have what? To be carboxylase. Right? So that's exactly what we're going to have. And don't forget, carboxylase is actually what? Of two types. We have A and also what? And also B. So what happens is that this particular enzyme, with this particular pancreatic enzyme, they actually have their let me say they have their target amino acid. You understand? They have their target amino acid. For example, trypsin basically target was the lysine or what? Or arginine residue. Why was the carboxylase A target was the aromatic what? Amino acids. We are not going to explain what the spectating was in this video. So basically, the trypsin, chymotrypsin, elastase, and carboxylase will act on what? On chyme. And don't forget, chyme contain what? The mixture of all these polypeptides, peptone, tripeptide, and also dipeptide that has basically mixed with what? With the hydrochloric what? Acid. And this bicarbonate will basically what? Act on the chyme, neutralizing what? The acidic content of what? Of the chyme, right? Making the action of what? Of all these enzymes optimal. Because this enzyme actually requires what? A pH that ranges between 7 to 8. So this bicarbonate will help us to neutralize what? The chyme, right? Thereby what? Making the product, which is the chyme product that is available in what? In the intestine. It will make it a little bit what? Alkaline, right? So at the end of the day, the trypsin, chymotrypsin, elastase, and also carboxylase will act on what? On this polypeptide, converting polypeptide into what? Into basically we're going to have some kind of what? Of tripeptide, right? Tripeptide. Then at the same time we're going to have what? They're going to act on what? On peptone, converting to what? We're going to have, let me say, dipeptide. Right? We're going to have the dipeptide, and at the same time we're going to have what? Amino acid. Right? We're going to have amino acids, some small, let me just say similar what? Amino acid. Why? Because the pro-carboxylase will basically act on what? On the carboxy end of what? Of the protein, or let me say of the polypeptide chain. Let's say we have what? We have amino end here, right? Then we are going to have amino acid is here, amino acid is here, we have amino acid, then we have what? Carboxylic acid here, the carboxy functional group. Now what happens is that the pro-elastase can basically act at the end of what? Let me say at the carboxy what? End. So once they cleave what? The carboxy end, what are we going to have? We are going to have what? Free amino acids. Right? Once they act by cleaving what? The let me say the carboxy end, we are going to have what? Free amino acid that can be what? Absorbed directly into what? Into the what? Systemic what? Circulation. So these are what? The amino acid that we have, right? Amino acid that are formed, or let's say that are produced as a result of what? Of the action of what? Of carboxylase. Now the chymotrypsin and trypsin, they basically target let me say their action is basically what? Inside. For example, if pro-carboxylase is acting at what? At the carboxy end, which is basically the first one as let me say exopeptidase. So pro-carboxylase is a type of exopeptidase because it acts at the beginning, at the carboxy end of the protein or polypeptide chain. Then from there, what will happen is that chymotrypsin, trypsin, and elastase, they basically act inside what? Inside the protein. That is, they basically act by cleaving up what? The protein. So the product is going to what? That is going to be formed as a result of their action. We are going to have what? Tripeptides, dipeptides, and let's say we are going to have some little bit of small chain, or let's say low molecular weight of polypeptides. That's basically what we are going to have. So in that case, polypeptide, tripeptide, and so dipeptide are formed, or let me say they are produced as a result of the action of trypsin, elastase, and chymotrypsin. But when it now comes to pro-carboxylase, the action of pro-carboxylase is actually what? The carboxy end. So in that case, if it's cleaved off the carboxy end, we are going to have what? Free amino acids. So that's why we are having what? Amino acids here. So from there, so after the action of the let's say of the pancreatic enzymes in the duodenum, then the product we're going to have is going to be polypeptides. Let me say low molecular weight of polypeptide, like low molecular weight. We can have it to be, let me say, tetrapeptides, but they are going to be very, very low. So we're going to have tripeptide and so dipeptide, and also including amino acids. So in that case, let me just simply rub this guy up because like calling it polypeptide simply means it sounds to me as if I'm talking about very, very high molecular weight. But let's just simply say we are going to have the product with tripeptide, dipeptide, and so amino acid. So that's going to be the end product of what? Of the action of the pancreatic what? Enzymes. Now we move on to what? To the inner part of what? Of the intestine, which is actually what? The jejunum. Now listen. So after the action of what? Of the pancreatic enzymes in the duodenum, then we move on to what? We move on further into the what? Into the intestine, right? Into the small intestine. Now let's say this part is actually referred to as the jejunum. Let me basically label it. Let's say this is the jejunum. Is another part of what? Of the jejunum. Is another part of what? Of the small intestine, right? Now what happened is that here we have some you know, at first I said intestinal cells are referred to as the enterocytes, right? Now every time you are saying here, these are referred to as microvilli. They are basically the structure, or let's say they are like how can I call it? They are just like finger-like structure that helps in the absorption of what? Of protein. Now amino acid is the small is the simpler form of protein, so it can be absorbed directly. Then this tripeptide and what? Dipeptide, they need what? Further digestion, right? Some are also what? Absorbed directly into the what? The intestinal cell. But one thing I want you guys to understand is that the intestinal cell, which is the what? The enterocyte, right? The enterocyte of the jejunum, they are going to release a special type of enzymes in this case. So in that case, these enzymes, we are going to have them to be amino. We are going to have what? Amino peptidase, or peptidases. Now at the same time, we are going to have another enzyme. We are going to have what? Dipeptidase. You understand? Then in some cases, we are going to have what? Tripeptidase. But basically, let's talk about dipeptidase and also what? Aminopeptidase. So what happened here is that these particular enzymes that you are seeing here, they are released by the intestinal cell, which is what? The enterocytes. Now what happened is that these dipeptidase will basically act on what? On tripeptide or dipeptide, right? Converting them into basically, we are going to have it to be converting them to amino acids. Look at this. We are going to have amino acid here. So these are amino acid. Then this aminopeptidase is going to act by cleaving off. It's just like it's going to cleave off what? The amino acid. Like for example, let's say we have a dipeptide and this is the dipeptide, right? So aminopeptidase will basically what? Will basically cleave off what? This particular what? Dipeptide into what? Into amino acid. So you're having amino acid here. So basically the name of this guy, I'm going to call it what? Aminopeptidase. Right? It is found what? In the bruised border, or let's say in the lumen of the intestinal cell, which is what? The enterocytes. Then from there, let's say we have what? We have a tripeptide here, or let me say a little bit polypeptide here. So one thing that happens is that dipeptidase can basically what? Is going to cleave off and produce what? A dipeptide. Right? It's going to cleave off the protein, act on the protein, and produce what? A dipeptide. We are going to have a dipeptide here. Right? This is a dipeptide. We're going to have, let me say we have amino end and so what? Carboxy end. So this is a dipeptide. Then from there again, we can have what? Tripeptide as well. It's going to cleave them off into what? Into three parts. Let's say we have another long chain of polypeptide here, right? So this long chain of polypeptide. Then in the case of tripeptidase, right? It can basically what? Cleave off. Like it's going to cut off this side and produce what? Three peptides, right? Three amino acid, which is referred to as tripeptide. So this tripeptide, this what? This dipeptide, and this what? Amino acid, right? So that's basically what the end product of digestion of protein. So protein are broken down from protein itself, which is a complex polymer, into amino acid, right? Amino acid, dipeptides, and also tripeptide, which can be absorbed into the intestinal cell, right? So the intestinal cell, the part of the intestinal cell of the enterocytes that are actually responsible for the production of let me say dipeptidase, aminopeptidase, and also in some cases we have tripeptidase. It's actually called the bridge border. So in the bridge border membrane, that's basically where dipeptidase, aminopeptidase, and so tripeptidase, that's where they have. And they cleave up they cleave up complex polypeptide or polypeptide with low molecular weight. Which can be tripeptide, it can be dipeptide, it can be amino acid. Amino acid will get absorbed directly into the enterocyte. Then from there, dipeptide will be acted upon by aminopeptidase or basically dipeptidase. Dipeptidase will cleave dipeptide into amino acids, right? Then aminopeptidase will start chopping off, start cutting them at the end, right? Start cutting them at the end. That's aminopeptidase. and then the result of action of aminopeptidase is going to be amino acid, free amino acid, and that's basically what you're saying here, amino acid. So that's basically what the results of the action of what of aminopeptidase, which is what we are saying here. But in case of dipeptidase, it will basically act what on the polypeptide, cutting them what into two amino acid, which is what dipeptide. It's going to cut them what into two peptide, like two peptide into a peptide rather. You understand? Which contains two amino acid. We're going to have two amino acid. That's basically what we have. So and this is what we are saying here. So this is dipeptide. We're going to have this what to be dipeptide, right? And this what to be tripeptide. So this is actually what the end product, or let me say the end product of the digestion of protein, right? So I'm going to talk about the absorption process in just a moment, right? And then if you enjoy this video, don't forget to like, share, and subscribe. So I'm going to be releasing the digestion process in another video, because this video is actually what long enough for the absorption. So let's quickly what recap everything that we actually said this morning, that we've been saying this morning. Then from there, I said once you consume what a high rich what protein, right? A food that contains high amount of protein, which can be meat, fish, and beans, right? Once you consume it, due to the action of the teeth, right? The teeth is going to is going to chew it. So once it chew it, the food is going to mix with the salivary gland, right? And form a bolus. Digestion of protein doesn't take place in the other cavity, it doesn't take place in the mouth. So the bolus will move down, pass through the esophagus, is going to reach the stomach, right? So once it's reached the stomach, then the stomach is going to have two special cells, the chief cells and also the parietal cell. The parietal cell will secrete hydrochloric acid, and at the same time the chief cell is going to secrete pepsinogen, right? So the action of pepsinogen is basically the pepsinogen that you're seeing here is actually in its inactive form, right? Why it has to be converted to the pepsin, which is the active form. So the parietal cell, which secretes the hydrochloric acid, the hydrochloric acid will help to convert pepsinogen back into pepsin, which is the active form of the pepsinogen. Then from there, the hydrochloric acid can also help to denaturalize the protein, right? It will act on the protein and denature the protein. So once it's denatured protein, just like it is unraveling the quaternary, the tertiary structures, it will unravel them, so that pepsin can actually act on it optimally. Now what happens is that once the hydrochloric acid converts pepsinogen to pepsin, pepsin can also go ahead and act on pepsinogen, converting to pepsin. Right? So once more pepsin is formed, then pepsin is a molecular cell, right? It's going to cut off basically the protein. It's going to cut them off. Then we're going to have these products. We're going to have peptides, peptones, tripeptides, and also dipeptides. So these are the results of the action of pepsin. And one thing I want you guys to understand is that pepsin is a type of endopeptidase. It basically acts inside the protein, like it acts on the protein inside. Its action doesn't start from either amino end or the carboxyl end, right? So it's type of endopeptidase. Then and it requires 1 pH that ranges from 1 to 3. So the stomach is highly acidic. So the acidic content of the stomach will mix with the polypeptide, peptone, tripeptide, and dipeptide to form a special kind of product here, which is the chyme. So this chyme is highly acidic. So once the chyme reaches the first part of the intestine, which is the duodenum. This is the duodenum. So once the chyme reaches the first part, it's going to act on some kind of endocrine cells of the intestines, which is of the duodenum. It can be in the duodenum or in the jejunum. So what now happens is that once the chyme is going to stimulate the eye cells of the duodenum to secrete CCK, right? Cholecystokinin, right? And at the same time, it's going to stimulate the S cell to release secretin. So once secretin is released, it will travel to the pancreatic daughter cells, right? And cause the pancreatic daughter cell to release bicarbonate. Then the eye cell, the CCK, will travel to the acinar cells, causing the acinar cells to release digestive enzyme. Right, digestive enzyme actually require bicarbonate for them to perform their function optimally, because bicarbonate will help to neutralize the acidic content of the chyme. It will neutralize the acid, making the pH of the chyme to reach 7 to 8. And that particular pH, that's where the digestive enzymes that are found in the pancreatic juice can perform their own function optimally. Then the enzymes that are found in the pancreatic juice, there was trypsinogen, chymotrypsin, proelastase, and procarboxylase. Right, so they are in their inactive form. they have to be converted to their active form. Then they will be acted upon by what? By a specialized enzyme here that is released by what? By the enterocyte. So this enzyme is called enteropeptidase, or simply called it what? Enterokinase. So this enzyme will act on trypsinogen, converting to trypsin. Then trypsin will in turn convert all these zymogens, convert them to their active form. Trypsin will act on chymotrypsinogen, convert to chymotrypsin. Trypsin will act on proelastic, convert to elastase. Trypsin will act on procarboxylase, converting to carboxylase. Trypsin will also act on procarboxylase A or B, converting to carboxylase what? A and B. Then the final product of this enzyme that we are having here is going to what? Tripeptide, dipeptide as what? Amino acid, because trypsin is going to act on what? On this peptide that you are seeing here, converting them to what you are seeing here. And then the action of trypsin is kind of what? Or specific. Trypsin acts on the lysine and also the adenine residue. Chymotrypsin was basically acts on the it basically targets what? Neutral or what? Aromatic amino acid. You understand? It's going to what? Act on them. Then from there, elastase like carboxylase will basically what? Target what? Aromatic what? Amino acid and clean them off at the carboxy end. That's basically the function of carboxylase. Then elastase also targets what? Neutral amino acid like what? Like glycine, any neutral amino acid. That's at the same time to have what? Chymotrypsin. At the same time, it does that as well. It targets what? Like neutral amino acid as well. Then from there, the action of this, or let me say the product of this the product that we're going to have due to the action of this enzyme is going to tripeptide, dipeptide, amino acid, and some low molecular weight polypeptide. Right? Low molecular weight polypeptide. That's basically what we are going to have. So once the food, or let's say the product, reach the jejunum, or let's say reach like further move into the intestine, then they are like when it gets to the bruised border of the intestine, rather. Let me just use the bruised border. It's actually making sense, right? Once it reaches the bruised border, what's going to happen is that the bruised border is going to release some enzyme. We have the dipeptidase, aminopeptidase, and also what? Tripeptidase. So these enzymes basically are going to act on tripeptide, dipeptide, amino acid, low molecular weight polypeptide. Let me just include this. We have low molecular weight polypeptide. Let me just put here polypeptide. Right? So these particular enzymes are going to act on them. They're going to act on tripeptide, dipeptide, amino acid, low molecular weight polypeptide, right? Converting them to amino acids. We're going to have amino acid, which is what we are saying here. Amino acids. We're going to have what? Dipeptides. We're going to have what? Tripeptides. Right? So that's basically what the end product of protein digestion. So this end product will then be absorbed by the enterocyte into the systemic circulation. So I'm going to explain that one in a separate video. So if you enjoyed this video, don't forget to like, share, and subscribe. Then the next video I'm going to make it